With that, I will start to introduce our next keynote. And th this next talk is actually quite a special one for us at BSI as well, because it's the first time we've had a repeat speaker. Cesar gave a talk at BSI years ago when I believe I met him through the Infectious Disease Consortium um, in Boston. And it's really wonderful to see him come back, not only because we've been following his career and he's been doing really incredible work across AI-driven antibiotic discovery, among many other uh, important additions to our knowledge, but also because of how involved he's remained and how committed he's been to diversity in science and promoting trainees from all different walks of life. With that, if Eugenio is online, actually, he will help me co-introduce Cesar and can give some more of the background. Thank you, Cesar, for joining us again.
Yeah, thank you.
Thank you, Payam. So let me introduce Dr. Cesar. Cesar de la Fuente is a Presidential Associate Professor at the University of Pennsylvania, where he leads the pioneering machine biology group and is among the youngest faculty members ever tenured at Penn Medicine. Trained at MIT and the University of British Columbia, his work centers on using computational tools and artificial intelligence to revolutionize antibiotic discovery and biomedical innovation. De La Fuente's team developed the first computer-designed antibiotic shown to be effective in animal models, helping establish AI-driven antibiotic discovery as a new scientific field. His lab has uncovered more than 1 million antimicrobial compounds by mining vast biological datasets. These advances have transformed the pace of antibiotic discovery, reducing processes that once took years to mere hours. Widely recognized for his impact, De La Fuente has received over 80 awards, including distinction from MIT Technology Review, the National Academy of Medicine, and the Princess of Girona Prize. He has published roughly more than 200 papers in leading journals and delivered hundreds of invited lectures worldwide. Additionally, advances from his lab include reprogramming venoms in antimicrobials, developing autonomous nanorobots to treat infections, creating novel resistance, proof of antimicrobial materials, and um, inventing rapid low-cost diagnostic tools for COVID and other infections. Please let me join in welcoming Professor Cesar de la Fuente. Thank you so much.
Thank you so much, Eugenio. Let me just share my screen. Can everyone see that? It's all right?
Yes, it looks
Yeah.
perfect.
Okay, perfect. Well, yeah, thank you for that very kind introduction, and thank you for inviting me back. It's a privilege to be one of the few that has repeated. And thank you for putting together such a wonderful conference to bring people together from many different places around the globe. I think it's a great initiative. So today, what I thought I would do is tell you a little bit of the work that we're doing, trying to develop computational methods to accelerate discovery in biology. And I guess the way we think about all of this in my lab is that we think of biology as this incredible source of technologies, right? If you can learn from first principles how some of these biological processes work, we might be able to then teach computers some of those principles so that then machines can just accelerate the pace of discovery. And so that's a lot of how we think about problems and so that's kind of what I'll talk about today. So we're really passionate about this chains, small or short chains of amino acids called peptides or small proteins. And at a high level, the way we think about this is that these are amongst the smallest biofunctional molecules of life, and they've been around forever, and they can actually execute functions in biology, right? And so if we can learn how to program them, we might be able to tackle many problems, not only in biotechnology and medicine, but also in sustainability and many other things. And in fact, polypeptide chains can be can operate as semiconductors and can be used for carbon capture. So it's not only within the realm of medicine, but the applications, I think, here, if we learn how to really program them in a predictable and reliable way, I think it can open up entire new avenues for advances in science. So a lot of what I'll describe today is around, learning how to do peptide design, essentially. We think peptides are an ideal training ground for AI research. They're very small. They have an astronomical sequence space. Because of their size, we can experimentally make them in the lab with robots, with solid phase chemical synthesis robots, and we can pretty easily scale. They're also very tunable. So if you change the optimization function, you can program them or design them to do different things, right? Like kill bacteria, but also maybe modulate immune response or target tumor cells. So really, the umbrella of functions is really incredible, and so I think we also see this as a big opportunity. Our proving ground for peptide design has been AMR, or antimicrobial resistance. And the reason for that is that I think this is really the greatest existential threat to humanity. And these are bacterial infections that are, highly resistant to everything that we have available today. So all antibiotics that you can think of. And in fact, these infections are associated with about 5 million deaths per year, every year, around the globe. And the projection is that if we don't come up with solutions by 2050, that number is going to double to 10 million deaths per year, becoming the number one cause of death in our society, including even surpassing cancer. And so I think this is a tremendous problem that demands urgent solutions. And so we're trying to do something about it in my lab. Compounding these issues, it takes a long time to even discover preclinical candidates. It can take 6-7 years to identify molecules that might be promising, and it's really expensive, right? These are U.S. dollars, but over $2 billion U.S. dollars to come up with something useful for humanity. So in an attempt to try to address this, years ago, we proposed that perhaps AI systems could be used to accelerate the pace of discovery in this field. And just so you get a sense, the way that the traditional paradigm of discovery is literally scientists going around nature, digging into soil or water, and then trying to identify active compounds, right, in that really complex organic matter. And as you can imagine, that is a process that relies on trial and error and is very painstaking, and it can take many years, right, with a very low hit rate. So instead of doing that, we decided to think about this problem a little bit differently, and we decided to think of all of biology as an information source, a bunch of code that could be mined with with the right algorithms, and so we decided to think of biology as information, and realizing that, of course, we had at our disposal a lot of biological data in the form of genomes and proteomes and metagenomes that we could sort through. So that was sort of our hypothesis, and so essentially we wanted to mine the code of life for new functional molecules with antibiotic properties, right? Knowing that if we could establish a paradigm for this, then we might be able to identify other types of functional molecules within the code of life, right? That do other things other than kill bacteria. So I'll go a bit chronologically how we did this. We started by using very simple scoring function algorithms that essentially recognize patterns of amino acids that are predicted to be antimicrobial. And so we started through a number of collaborations by mining proteins to see if we could find amino acid fragments hidden or encoded within the protein that would have these antimicrobial properties. So a protein, you can visualize it in 3D, but also in 2 dimensions with the amino acid code here. And these systems, what they do is they run through the code and they identify regions that are predicted to be antimicrobial. and those are given by these different colors delineated here. This is only a prediction, but it can be quite powerful, as we found, because it allows you to identify potential antimicrobial fragments in proteins. For example, this fragment in yellow here. And then utilizing synthetic biology methods, we can actually extract that fragment from the protein and we can try to develop it into something that looks like a therapeutic. So we did this for a number of projects, mining one biological system at a time. But then with advances in compute, we realized that we could scale up. And so we decided to embark on this journey to try to, instead of, let's say, doing one protein at a time, doing this in a massively parallel manner. So the idea here would be to utilize our sort of pattern recognition algorithms as barcode scanners that would scan through many, many proteins to identify barcodes that look like potential antimicrobial sequences. And so this was the concept. This was the hypothesis. And to try to test this hypothesis, we mined the human proteome for the first time in a systematic way as a source of antimicrobial compounds. But this really imposed a big scaling sort of challenge for us because, as I mentioned, we were used to mining one protein at a time, or one biological system at a time for antimicrobial discovery. And here we had to scale that up to 42,000 proteins. We actually went beyond the 20,000 proteins encoded in our genome, and we took into account isoforms. So it was a huge scaling problem, really. And what was incredible is that it took just 1 hour on 48 CPUs at the time to be able to kind of run through the human proteome and identify a bunch of new potential functional antimicrobials. From a biological perspective, this enabled us to find a whole new world of antibiotics that we call encrypted peptides. So thousands of them encoded in our genome and our proteome. And so out of those thousands, then we did experimental validation on 56 of them. But before we did that, we actually we did an analysis on a lot of these sequences that we found. And what was striking is that the vast majority of these fragments are derived from proteins not involved in the immune system. So 98.3% come from proteins that don't have a predicted or real function that is associated with immunity, and only 1.7% actually come from host defense and immunity proteins. So this was quite striking to us because you tend to think of antimicrobial molecules as being associated with host immunity and immune proteins. And so this led us to propose this hypothesis that we call the encrypted immunity hypothesis, where we think that, or we propose that there might be hidden layers of immunity that are kind of encrypted or encoded in our biology that only get unleashed or released upon certain contexts, like the context of an infection or stress or things like that. And so this is a working hypothesis or working model that we're continuously testing, but there is increasing evidence from us and other groups that this hypothesis might be going in the right direction. So it's exciting to think of biology as this encryption system of information that is not only monodimensional, but there may be many dimensions that can get decrypted when needed. Like I mentioned, we synthesized 56 of these peptide molecules using chemistry, solid phase chemical synthesis. We did experiments with them to see if they could kill bacteria in the lab. And they had this broad spectrum activity, so they could kill both gram-negative and gram-positive pathogens, which was interesting. And then through other machine learning methods that we've developed, we saw that many of them, or some of them, are predicted to be cleaved off by proteases from the parent protein where they come from. And so we believe that that's one of the mechanisms of activation of these encrypted sequences is through proteolytic cleavage. There may be other mechanisms of activation. We then went on to show that some of these encrypted molecules from the human proteome could actually reduce infections in mice, clinically relevant infections caused by gram-negative pathogens like Pseudomonas and Ewamani. So here we see the untreated control groups in gray, and then when we treat them with one encrypted peptide, you see a decrease in the infection with a different peptide here, and then in purple we have a cocktail of these two together. And we typically see synergistic or additive interactions, which recapitulates what we see in vitro, also when we do synergistic interaction assays. So this is kind of now creating a conceptual model where we think that upon infection or upon different physiological stressors, perhaps some of these peptides are getting released, and they actually work together in synergy to mount an attack against invading pathogens. We've seen that some of them are expressed physiologically at different levels between low, medium, and high, and in different parts of the body, like I mentioned, including in the nervous system, the cardiovascular system, the digestive system, and many body systems that are not typically associated with immunity, right? So I think this is a way of connecting all of it, and connecting our whole physiology to achieving host immunity, which in the end is if you think about it, is probably the most important thing for our survival day to day is that our immune system is working well. And if you think about it, it's actually quite amazing that we don't die every hour because we're being exposed to millions of microbes all the time. And so it's almost a miracle that we don't get sick more often. And then more recently, we published this paper where we synthesized a lot more of these molecules from the human proteome, and we saw that they, in addition to their ability to kill bacteria, they could also modulate the immune response and balance inflammation. So the journal kind of highlighted that these peptides might be a hidden layer of immunity, and so kind of that's the working hypothesis that we have. A lot of this work really sparked a lot of new ideas and in my lab, and one of which was that we proposed that perhaps these encrypted molecules were conserved throughout evolution and across the tree of life. And to try to tackle that question, we decided to look at our closest ancestors, Neanderthals and Denisovans, to see if we could find similar encrypted sequences encoded in their genetic code. And in order to tackle this, we brainstormed a lot about how to do this, and we came up with this conceptual framework that we call molecular de-extinction, whose ultimate goal is to identify functional biomolecules. This can be, peptides or small proteins or metabolites that were produced throughout evolution, and then with the goal of synthesizing them in the lab throughout evolutionary time to try to learn how mutations that took place in those molecules influence function, which can be, an ability to kill bacteria or target tumor cells or boost immunity, you name it, really. We're not restricted. So this is a conceptual framework for thinking about sequence to function across evolutionary history. Going to the ancient hominid work to try to do this, we actually developed this machine learning pipeline that we call Pancleave, that at a very high level, what it does is it takes all the proteins available from, let's say, Neanderthals. It chews them up in little fragments by predicting the cleavage sites on each of the proteins. And then here, once we have this soup of fragments, we run machine learning filters to predict their antimicrobial activity. So this pipeline enabled us to discover what we think are the first potentially therapeutic molecules in ancient proteins. Here we have examples of Denisovan proteins and Neanderthal proteins. We use the modern human proteome as a control in this sort of computational work. And then of course, this is all predictive work. So then what we did is we synthesized using chemistry a lot of these peptides, and we did experiments in the lab, in the real world, to try to learn from them. So some things that we've learned is that the modern peptides tend to have a different mechanism of action of killing bacteria compared to the ancient ones. So if you think about a gram-negative bacterial cell, you have two membranes. You have the outer membrane and the cytoplasmic or inner membrane. So what we saw is the modern molecules tended to go after the outer membrane, whereas the more ancient ones tended to target the inner membrane. So I think this is an illustration of the potential that we see in applying this framework, where we can really unlock new biological insights, perhaps, and learn more about how evolution did things, and perhaps try to begin grasping the evolutionary reasons of why certain things took place at certain moments in evolution. We then, for the most promising candidates, we took them all the way to the preclinical stage in different mouse infection models, including a skin abscess model and a deep thigh infection model, which is the one that the FDA here in the US typically wants to see for exciting preclinical candidates. And what we observed mostly was bacteriostatic effects in these mouse models. So basically, when we treat the mice with some of these encrypted molecules from, let's say, Neanderthal, the Neanderthal genetic code, they can contain the infection, but they're not actively killing the bacteria. And so none of these are going to be an antibiotic tomorrow. But nevertheless, this demonstrated to us that it was possible to first dig into the genetic data of ancient organisms, identify functional molecules, and then bring them back. And we've seen that they have practical applications, right? Even at the preclinical stage. The best molecule that we found here was we call it Neanderthalin, and it comes from the Neanderthal proteome. And so these results actually gave us a bit of more confidence and to ask a more ambitious question, which was why don't we try to scale this up once again and mine through all of ancient biology, if that's even possible technically, right? So we went back to the drawing board, and we, as it turns out, all of ancient biology is quite a lot of data. So we had to basically develop a more powerful AI model to be able to do this in an efficient way. And that's when we developed Apex. This is its architecture. It uses our RNNs plus attention neural nets. And from a practical standpoint, what Apex allows you to do is you as an input, you have an amino acid sequence. And from that sequence alone, it's capable of predicting the antibiotic function. So it's a sequence to function prediction model, which is quite hard to actually achieve. And Apex enabled us to take a journey through evolution and dig into the proteomes of ancient organisms at a scale that had not been done previously. And here are ancient organisms and real peptides that we found in each of them that are predicted to be antimicrobial. Here we have an illustration in this schematic of this journey through the Holocene and the Pleistocene. And here I'm showing sort of illustrations of some of the organisms from whose proteomes we found some of the lead candidates. So it includes ancient penguins that were extinct in the 50s. It includes ancient zebras, magnolia trees that disappeared throughout evolution. It includes ancient elephants, woolly mammoths, giant sloths, and many, many other creatures that used to roam around our planet, but unfortunately, through different extinction events through evolution, they disappeared. But what's really interesting here is that they disappeared as species, but we have access to some of their biological code, right? And so now with algorithms, we can explore that code to try to find interesting things and potentially functional molecules, but also to try to learn from them, right? So the way we think of this is we think of molecules as documents of evolutionary history, and where we can learn about how they evolved, how they changed, and why that happened. What were the conditions that that made those changes, that triggered those changes, right? So here we have all of ancient biology captured in a single image. I never thought we would be able to do this, but it took a long time. But so you can get a sense of the scale of the exploration enabled by Apex. These are all the organisms whose proteomes Apex was capable of exploring. And we have both predicted encrypted peptides or EPs and active and predicted by Apex, of course. And then whenever it says active, we actually synthesize all of those peptides using chemistry. And and we tested them in the lab for antimicrobial activity against different pathogens. And so whenever it says active, it's actually ground truth data from the lab. And we've done uh, this kind of scale up of exploration for functional antimicrobial molecules, but we believe this proposes a paradigm where you might be able to do this for anti-cancer molecules or immunomodulators or many other things, right? Simply by modifying their objective function. A lot of the lead compounds, we took them all the way to the preclinical stage, again, in the abscess model on the top and the deep thigh infection model on the bottom. And in this case, we were able to find highly bactericidal molecules, including mammuthicin 2 from the woolly mammoth and other compounds from the ancient sea cow, giant sloth, and other organisms that exhibited anti-infective properties that were comparable to polymyxin B, which is a last resort antibiotic, right? So another thing that we learned from this experiment is that if you scale up how much biological data you're exploring, you're more likely to have a higher content of higher yield of functional molecules, at the end of the experiment. So this, I think, is another lesson of the things that as you scale your exploration of biology, it actually it gives you a lot more in terms of your particular objective function. And this is actually, we had proposed this standardized data set hypothesis a long time ago, but now we have actual empirical data where with increasing training set of molecules, so these are experimentally determined, and the whole premise of this hypothesis is that if we generate experimental data in the lab that is high quality, that is standardized, and we train models using that standardized data, the models will become, there will be a scaling law in the sense of how reliable and accurate the models will become. And that's what we're seeing here. So this is accuracy of the model Apex over time as we train it using more high quality experimental data. And this is hit rate as determined experimentally, right? So we actually synthesize things and we test the accuracy of the model. And as you can see, it's almost, it's around 95% now accurate. I do think that for each narrow task, so in this case, obviously it's predicting antibiotic function, and now we're working on many other tasks in parallel. If you get to 95+% accuracy, you can really rely on the models. You can trust them, and I think this will lead to us having to do a lot less experiments in the future. Once we can train models that are this good in biology, which is it's non-trivial, it's hard, and typically I think at least for now is going to be on narrow domains like predicting antibiotic function, predicting toxicity and stability, folding, things like that. So we think of biology as information in this emerging field of antibiotics AI and other fields. And so one of our premises is that we wanted to mine all of it to try to find functional antibiotic compounds. So one of our early missions or dreams was to mine across the whole tree of life, which has three primary branches, as we can see here: eukaryotes, archaea, and bacteria. And a lot of the initial emphasis, as you've seen, was in eukaryotes, where we belong. But then we also were really curious to explore the bacterial world and the archaeal world, which are two huge branches of the tree of life. So I'm going to briefly mention some collaborations where we tackled the bacterial life to try to digitally mine it, to try to find antimicrobials. This is a collaboration with Luis Pedro Coelho and his lab, where we use machine learning to mine through what we call the global microbiome, which are over 60,000 metagenomes and almost 90,000 microbial genomes that come from all around the planet. It's really a great representation of the microbial life on Earth. And I think it was really exciting that we were able to identify almost 1 million new antimicrobial sequences by doing this exploration digitally. And we've released all of this out to the world through this website that is open, fully open and free access. So anyone has access to all these potential candidates, and anyone can develop them further. And so we did this in an attempt to kind of accelerate progress in this area, in this really dire area of antimicrobial resistance, where we really urgently need new innovations and new solutions. The other collaboration, another wonderful collaboration with Amy Batt and her lab at Stanford. Here we mined human microbiomes. As you know, these are bacteria that live in and on us. They're good for us. They can live in our mouth, skin, urinary tract, intestines, and so on. And the idea here is that, of course, these are good microbes for us, but in their native environment, let's say the intestine, they still need to grow and sort of outcompete other surrounding microbes. And so we thought maybe they use these encrypted peptides or peptide molecules as a form of chemical warfare to enable them to reconfigure those microbial communities around them. And at least in some cases, we did find that individual microbes were producing individual peptide weapons to kind of kill surrounding microbes. And so we think this may be another part of the toolkit that microbes utilize in the context of their native microbial communities to kind of reconfigure their surroundings and also influence the ecology of their communities over time. And in this case, we also found this lead compound in infection models, prevotellin 2, produced by the gut microbe Prevotella copy, that was highly effective in a number of different infection models. And more recently, we were able to kind of complete this exploration across the tree of life by mining, by digitally mining the biological data available for archaea. And archaea, I just want to say that they're these really esoteric organisms that for the longest time we thought that they were bacteria, until really pioneering work by Carl Woese and others by sequencing the ribosomal RNA of these organisms, figured out that they actually belong to their own branch of the tree of life, which was a pretty incredible, I think, discovery. So here we leverage a retrained version of Apex. So this is actually Apex 1.1, where we train it on more experimental data, high quality data. And so it's sort of an improved version of our Apex 1.0 model. It was able to mine the archaeal data and uncover archaeocins that are encoded in archaeal data that are highly effective against clinically relevant pathogens. And so this really sort of completed this exploration, this early mission that we had in the lab to kind of do that at scale. So sort of to summarize a lot of this work, with traditional methods of antibiotic discovery, it can take many years to identify potentially preclinical candidates. But now with the computational methods we and others have developed, we can do this at digital speed. And so in a few hours, we can discover hundreds of thousands of candidates. And if we really push our resources, we can synthesize them, not all of them, but some of them, and test them in vitro within 1 or 2 weeks. So it's really this amazing playground for a scientist like myself. I've been dreaming something like this for a long time, and now we actually have this playground where we can integrate into our workflows and our experimental setups these algorithms that are increasingly more reliable. We can trust them more, and that are trained on really high quality data that is experimentally generated, right? I think that's an important part of the equation, that is not only the AI systems and machine learning, but also the data that we use to train those systems that is crucial here. So what next? So I'm just going to briefly mention a couple of things that are in the pipeline. This is Apex Go, stands for generative optimization. So this is a generative AI model to optimize for function. So as an example, you can start with a template. This can be, for example, a peptide template that is a lousy antimicrobial. It has a very high MIC value, meaning you need a very high dose to actually kill bacteria. So it's a really bad antimicrobial. And through generative Bayesian optimization, what we're trying to do here is we're trying to generate synthetic, new to nature peptides that nature has never thought of, that are predicted to have a lower MIC value, right? So we're trying to minimize the MIC number to try to find potent antimicrobials. So all these dots that you see here, they're generated by the model. And what we then did is we synthesized a lot of these and we did experiments. We found that 85% of them were capable of killing bacteria in vitro. But more importantly, 72% were better than the template at killing bacteria. So I think this is the important metric here, where this is the optimization, right, of the initial template. We've done this to try to minimize the MIC, but of course you can imagine you can use similar systems to minimize or to optimize for your particular objective function. If you're interested in targeting tumor cells or diminishing inflammation or boosting immunity, we think of this as a potentially it can be extrapolated to other functions, right? And this correlates in animal models too, where the synthetic peptides that are optimized are better than the templates at reducing infections. We're also interested in building multifunctional molecules that can do more than one beneficial thing. And here we decided to apply this concept to a big problem in infectious diseases, which is the problem of intracellular infections. These are bacteria that enter into human cells when they infect us, and they are really kind of smart in the sense that they hide inside our cells in a way that the immune system cannot really kill them, and conventional antimicrobials also cannot reach them and target them. So we developed Apex Duo for dual function, which is a generative model that can design synthetic peptides that are predicted to do two things. One, penetrate into human cells, and two, find the bacteria and kill the infection, the bacteria that are kind of hiding inside the cells. So we generated a bunch of these synthetic peptides, we synthesized them, we tested them, and many of them were really good at killing bacteria because our models are really good at predicting that. But only a few were capable of penetrating into human cells, and only 3 were capable of doing the dual function. So the hit rate was quite low. We knew this was going to be a challenging project because, I mean, in the end, we're trying to endow a single molecule with two functions. But with those 3 that worked, to some extent, we were able to see a reduction in intracellular infections in human keratinocytes and human monocytes. And then we tagged them with a fluorophore Cy5, and we were able to do microscopy to kind of track their journey penetrating into human cells and then targeting not only Staph aureus intracellular infections, but also Listeria and Salmonella. And finally, Apex Oracle, this is one of our latest models. This is a multimodal model that bridges understanding between genomics, language, and chemistry. As the input, you have a genome of a pathogen and some associated traits with that particular genome. And then this Apex Oracle model can do 2 tasks. It can do a prediction task that predicts out of all the small molecules and peptides that it was trained on, which ones might be active against that genome of interest. And it has a generation task or generative AI task that can generate synthetic small molecules and peptides that are predicted to be active against that genome. The cool thing is that the prediction task takes seconds and the generation task takes 4 minutes. So we see this as a way of coming up with molecular solutions against future pathogens or emerging pathogens, right, in the context of infectious diseases. But even beyond that, if you think of in the context of cancer, tumors also evolve, and so you're always trying to catch up and come up with medicines and and molecules that are active against those evolving tumors. So in that context, we haven't tried it yet, but it might be useful in that context as well. And we're currently doing experiments on this. So the model works well, the performance metrics are good, and now we're trying to validate this in the lab. So I'll have more news later this year on its performance or its lack thereof. And with that, I'd like to thank lab members, past and present. Really fortunate to work with incredible people that come from many different backgrounds. Like we truly are a transdisciplinary group. And then our amazing collaborators from around the globe. We collaborate very extensively. Our funders, and I'll leave you here with links to our website, email, and social media in case anybody wants to get in touch. And this is the group, and I thank you for your attention and would love to take questions. Thank you.
Thank you so much, Dr. Cesar, for this wonderful and beautiful talk. So we have a
Thank you.
couple of questions regarding your thoughts about antimicrobial resistance. First of all, how do you predict, Eleanor asked, how do you predict the antibiotic function that will also evade antimicrobial resistance in your peptides?
Yeah, well, Eugenio, thank you very much. And so we are working on that. That's a challenging problem. I mean, I think Apex Oracle could help in that, right? In the sense that it can come up with solutions, at least computationally at the moment, as resistance emerges. But in addition to that, what we're doing, so Ben Sprung, a great postdoc in the lab, is actually working on an evolution project where he's forcing evolution by using a mute S hypermutant strain of E. coli, um, and he's exposing it to many different peptides that we have, and to try to figure out what the mechanism might be, what the targets might be of resistance. And so the idea of that project is twofold. First of all, identify new targets, learn how evolution evolves to some of these peptides, and then third, create a database. Actually, three points. Third, create a database, a training set that then we can use to train AI models to design what we call resistance proof molecules. Even though I know that that's impossible, but at least to try to minimize the selective pressure towards resistance as much as possible. So in the future, hopefully, something like that could translate in the clinic.
And regarding the antibiotic resistant bacteria, have you tested your peptides in clinical isolated bacteria that are relevant, like are common in hospitals?
Yeah, we have. So we've expanded a lot of our testing in vitro to some highly drug MDR multi-drug
Okay.
resistant strains, clinical strains. Yeah.
And yeah, we have one question from Brandon. He asked if it's feasible and worthwhile to scale this to also consider post-translational modifications in your encrypted peptides.
Yeah, great question. So we're currently working on that. So I don't have an answer yet, but we're working on a model to kind of integrate post-translational modifications as part of what we can do. We're also working on non-canonical embeddings for non-canonical residues, including D-enantiomeric residues. We obviously currently do that on the chemistry side, but not on the computational side as much. So those are all things that we're working on actively. Yeah.
And I also have a question regarding that you did this analysis for looking for these peptides in ancestry. Is there any particular time during evolution that you have more of enrichment of these peptides in some of species? And if you could also correlate this with the genome of the species.
Yeah, so what we're doing now that is sort of related to that question is so we have this hypothesis that there might be positive selection for some of these antimicrobial sequences. And so actually Ben, the same postdoc, star postdoc in the lab, he's actually doing that. And there seems to be interesting data so far. I mean, I don't have all the information yet. It's an ongoing project, um, but I think soon we'll have more conclusions on that, probably in the coming months.
And yeah, one more question from Eleanor. She asked if if you're planning to use these peptides to, like in another context, to selective reconfigure the microbiome in immunocompromised individuals, like to optimize their
Yeah.
immune response to other challenge rather than just infection.
Yeah, I think that's a really interesting question. We have a project on microbiome reprogramming peptide design, and we have some interesting data there where we can target microbes even at the species level and genus level, and not all the time. So it's not programmable yet, the pipeline that we have, but we can identify individual peptide molecules that can do such things. So that indicates to us that it might be programmable in the future as we understand more in depth how they work. But yeah, we're absolutely interested in that. We're interested in anti-inflammatory peptide design, designing them to boost certain cytokines and things like that. So we're really expanding a lot in terms of the application areas that we're applying a lot of the peptide design work that we're doing.
One more question, Dr. Cesar. Avida asked, while discovering antibiotics, major issue come antigen cross-reactivity with resistant development. How your APIX, how your APIX model rule out this problem?
Great question. It doesn't yet. That's something that we haven't done yet that I would like to do. I mean, we've done a lot of mouse work where we don't see adverse effects and things like that, but we haven't yet done the experiment to test whether antibodies get produced when the host gets exposed to some of these peptides. So we haven't done that yet.
Okay. Well, thank you so much, Dr. Cesar. It was an honor and a pleasure to listen to you.
Thank you for
And
having me. Yeah.
yeah, thank you. So I will go with Robin, who will introduce the next speaker.